So I had a couple people tell me they're here to hear my words of wisdom on subsurface phosphorus loss. I'm going to tell you right now, we do not have all the answers. And it's kind of interesting working on this issue. My predecessor, Tom Sims and Frank Cole and all those guys, they kind of got the runoff and the erosion thing down because that was the easy piece of the puzzle, and they left us with the hard piece. So I work with, this is the work of our group that's been looking at subsurface phosphorus loss for the last 3 or 4 years. I can't say that I did this alone. I work with some really talented individuals, including Tony Buda at the ARS, who's a research hydrologist, and Lee Slater at Rutgers University in Newark, who is a geophysicist. So what I'm going to present today is some pretty high-level geophysics and hydrology work, and I am neither a geochemist or geophysicist or a hydrologist, so I also have to digest this in a way that makes sense to me, so hopefully what I present to you will make sense to you too. I got some fancy pretty pictures, and that makes it really fun. You'll also see that Lauren Macesso is listed as a co-author on this talk. She's my PhD student, and she was really excited to see what we do here at crop school, so I said, if you'd like to come, then you can come and talk about what you're doing in the field. So she'll join us near the end of the talk. So real quick, why do we care about subsurface phosphorus losses? Anybody want to? Come on, it's the end. We got the bay. Well, partly because, especially on the Delmarva Peninsula, we have a lot of fields with artificial drainage networks. And we need these artificial drainage networks in order to farm a lot of our fields because of high water tables, seasonal high water tables that would prevent us from putting equipment in the field at the time we need to plant. What happens though is that we circumvent the natural hydrology of the system, and we actually move water through it much quicker than we would have if we weren't draining these systems. And in Delmarva, we are draining our landscapes mostly through open ditch networks. So all these little lines that you see on the slide here are ditches. This is actually interesting. I was pulling pictures off of this from Google Earth the other night, and they actually had this year, June of 2018, as the imagery. And the first thing I said is, why isn't it green? And then I remembered how wet it was, and most people hadn't gotten their crops in until real late in the season. But one thing I think was really stood out to me is that this is a fairly intensively drained area, and look how wet it still is. So even with our artificial drainage networks, we still happen to have a lot of moisture depending on the weather conditions. Ditch drainage isn't just an issue on the Delmarva, it extends far beyond nationally to some specific regions, like North Carolina, the coastal plain of North and South Carolina. You've got lots in Florida, and look along the Mississippi River here in Louisiana and Texas, and then up on that Wisconsin and, Minnesota and North Dakota border up there. So quite a bit of ditch drainage going on. We don't see as much tile drainage, but there is quite a bit of tile drainage in the United States too. This is actually a picture I took this past year in Ohio. We were doing a windshield transect looking at cover crop adoption. And it was really impressive, the number of tile lines that were going in up there. Driving around Wood County, Ohio, it looks exactly like Sussex County, Delaware. I thought I was still at home. But they have a very different reason for drainage. They have very, very heavy, thick clay soils. So when you look at tile drainage, this is what we mainly see around the Midwest. But drainage is drainage. You're circumventing that natural hydrology and getting water out of the field. When we're talking about ditch drainage on the Delmarva, there's been some work that was done in the early 2000s by the USDA that suggests this subsurface pathway is dominant in our fields. And when we look at some of our ditch drain fields, they are pretty much flat as a pancake. You go try to shoot a slope on there and you get 0.5% slope. So in this particular case, what we see is a lot of times that during the winter time, that water table is up real high, and about 90% of the water flowing into those ditches is coming from some sort of subsurface pathway. And if that water interacts with soils that have high phosphorus concentrations, then you get high amounts of phosphorus discharged to the ditches. Only about 10% of the water that we're seeing in these landscapes is moving via overland flow or surface runoff. And that's why we know that understanding what's happening under the soil surface in these subsurface hydrologic pathways is really important, especially if we're going to start looking at things like phosphorus risk loss in artificially drained fields. But again, like I said, it's not the easiest thing to do. So why do we still have so many issues or questions related to this subsurface phosphorus loss pathway? Is that work that they did in the early 2000s enough to say everywhere 90% of the flow is coming from below the surface? And we think not necessarily. Well, like I said, my predecessors worked a lot on surface runoff and erosion, things you can see, right? It's easy to measure the things you can see. You can set up little collectors at the edge of the field, and all the water that drains to the field can be funneled through these weirs, and we can collect the water samples and know a little bit about what's coming off in those surface runoff. Similarly, we can collect erosion that is moving over the soil surface in rainfall events. But it's really difficult to look below the surface unless you're like Sean Tingle and you have X-ray vision. I wish Sean was here because I think he'd enjoy that I've given him X-ray vision here. This is my joke for the day, so hopefully you liked it. We have a variety of different tools we can use to look underground, and most of them are quite invasive. So when we start working with folks like the USGS who are interested in groundwater flows, a lot of the way that we identify how the water is moving in the groundwater and what contaminants or nutrients are moving in the groundwater is based on well arrays. So they essentially go and poke holes at different depths and try to extract the water from those wells, and they measure things. They do carbon dating for age, and they measure nutrients and other types of things, turbidity, suspended solids. But depending on how deep you are putting your well is going to influence the properties of that groundwater. So we know that shallow groundwater tends to be younger, deeper groundwater tends to be older. So when you pull water from a well, you're pulling water that is maybe a mixture of ages. And so what's in that water is reflective of past practices that could be anywhere from 10 to 40 years old. And remember, we need to poke a lot of holes to really understand where the flow is going and understand what the concentrations of nutrients and other type of materials in the water are. My colleagues at USDA ARS have done quite a bit with intact soil columns. So here they are excavating. They've driven this box down into the ground. They then excavate the soil around it so that they can install their collectors underneath the boxes so that they're not disturbing the natural soil. They fill it back in, and then they can climb down in the hole and collect the leachate that comes out. So realistically, they're only looking at what's flowing vertically through a small amount of that soil, and look at how much disturbance they had to do to get that information. So it's not really feasible for us to go out and put a whole bunch of these intact soil columns around so that we can measure leachate. The other thing about these is they remove the ability to look at what's flowing laterally in these systems. They're only looking at what's draining through the soil vertically. Another way you can do this with the vertical is you can actually drive these in the ground, and you can pull them out and you can bring them to a greenhouse and leave them on a table. We actually have about 26 soil columns that were collected this way that have been sitting in our greenhouse for like 10 years, and they keep asking me if I'm ever going to use them. I tell them, please don't get rid of them. They're really difficult to take. So again, a lot of labor, a lot of disturbance, and not necessarily all the answers that we want. These photos came from my colleagues out at the USDA station in Columbus, Ohio, where they have much more tile drainage. And so this is typically what you see on their edge of field sampling sites. The benefit they have with tile drainage is they have a point source, they have a spout that you can then measure the water coming out of it. And you know where that tile is draining, so you can get a better idea of water flux, like how much water is moving through the system, and you can easily collect the sample and you can quantify nitrogen and phosphorus in those samples. But we have ditches, and ditches act a lot more like streams. In fact, a lot of our ditches are actually just channelized streams. The larger order ditches are streams that have been straightened so that we can more efficiently remove water from the fields. And what happens is you don't have a point of discharge in the same way that you get with a tile. What you have are seeps through the face of the side of the ditch. So this is a stream in Pennsylvania, but it illustrates the same point. And so here's our stream, and in the wintertime, my colleague Tony Buda was able to take this infrared camera out, and he can look at for temperature differences in the water that's in the stream versus what's coming in from the groundwater, because groundwater in the winter is warmer than the surface water. And so what you see in this picture is these blue colors is actually this stream water, and then this red, blue, and purple and yellow is warmer water that is seeping through the side of the stream bank. And so how do you measure phosphorus in this? How do you collect water that's seeping out of the banks of a ditch? So there's been some suggestion that maybe we can find these different seepage faces and we can install collectors that would allow us to drain some of the water from there and actually get some nutrient content. But this is incredibly difficult, and it would really take a lot of people smarter than me to figure out how to do it well. So again, not as easy as it is to go collect runoff from a variety of fields at the edge of field. So what instead we've done is we've relied on ditch sampling, and I want to thank Clint, who's in the audience, because he rocked me with this picture, and I can show you that we can set up these sampling stations. This happens to be one of the ditch filter sites that Josh McGrath had set up. We can measure water concentrations through these ditches similar to how we would measure surface water. But then we have to rely on this idea that 90% of the ditch water is related to subsurface flow and not overland flow. And that gets a little bit tricky because unlike a tile, which is draining a specific piece of the land, the ditch networks are connected. So you have upstream effects going on, so water that's come from other fields, water that's come from our field, and this idea that we have to believe that 90% of the water that's moving through these ditches is coming from subsurface pathways. So this has made it really difficult for us to do things like evaluate our risk assessment tools, like the P-index, the P-site index, or the PMT, especially when it relates to how we manage risk of subsurface losses. So we have almost no data on subsurface phosphorus losses, no load or concentration data. And when we start looking at the recommendations for how to evaluate a P-index, they want you to have 5 to 10 years of load data to compare your scores with. We know that it's difficult to measure, I just explained that, but it's also difficult to model subsurface phosphorus transport. So a lot of our hydrologic models rely on this idea that water flows downhill. And so when they run these models, they look at rainfall and other climate conditions, soil moisture, but they also need topography to move that water from a higher point to a lower point. And like I said, a lot of our fields, 0.5% slope, there's pretty much no way that that hydrologic model can account for that. And so what happens is when the smart people who do all the modeling try to get it to work on the Delmarva, it just doesn't work. It doesn't initialize and you get no data. So we can't use modeled data to help us evaluate this. And like I said, almost all of our data is short-term and almost all of it is column related, so vertical leaching only. And we know we have both vertical and lateral components at work in our systems. I was able to look at a little bit of data that we had, and I compared the subsurface piece of the PMT and the Delaware P-site index to our load data. So we had 6 drainage ditches with loads, and we had 8 leaching columns with leaching loads that I could compare to. And you can see they are all located on the Delmarva, mostly south of Sussex County and into Virginia. And this is what our results look like. So here's our dissolved P load, and I apologize, this was done for a scientific publication, so it's in metric units. But ultimately, when USDA says we should have a high risk for loss, you're up above this 2.5 to 3 kilograms per hectare. And you can see that in general, it looks like when our index score is low for subsurface phosphorus loss and our load is low, we pretty much come out here and it moves up where high is high. And so here's the PSI and here's the PMT. You'll notice that we don't like these regression equations. They don't do very well, right? There's only accounting for 26% of the variability. you can see right here, we way over-predicted the potential here, similar here. But I only have 6 points. How can I say anything about 6 points, right? I do not feel confident in this data to say, oh yeah, we're doing a good job. Similarly, when we look at the leaching scenario, it's a little bit better. We got 8 points. And we see that in general, when we have low load, we tend to have low risk. So high load, high risk. For PSI, much better correlation here, and the PMT. But look at these ones with the dots that are not filled in. This is where we had preferential flow. And we weren't able to determine the loads with the PMT when we had preferential flow. So what am I talking about with, oh, I'm getting ahead of myself. So I'm going to explain the preferential flow and matrix flow in just a minute. This does not even account for the fact that we have a lot of variability in our scoring between states. I'm not even going to touch on that other than to say that a lot of times we would score high for subsurface with the PMT and we'd still score low and allow N-based management with the PSI. So that's not anything you can even touch with what we're doing. Okay, so preferential versus matrix flow. There is nothing that we can do when walking a field to say this field is subjected to preferential flow. What we mean by that is there are cracks or large holes that move water through the soil vertically very quickly. These could be earthworm channels, they could be cracks in clay, they could just be larger structural pores that we have in the soil. Versus a matrix flow, which has to follow this more tortuous path between soil particles, and the water moves much more slowly. We can make a guess that if a field's been in long-term no-till for 20 years or so, that you're probably going to have more preferential flow, because something like tillage breaks up those large pores and large channels and makes it so that you have more matrix flow. But there's nothing in the soil series or the soil survey that says to us, this Pamunkey series is going to be preferential flow, or this Quindaqua series has preferential flow. We can't really look at the properties they give us and just decide that this is going to be the case. And it's going to have implications for how water moves in the subsurface. The second thing that we had a lot of problems with when we started looking at the P-index and the PMT and how we could possibly evaluate this tool was this idea of drainage intensity. So I think it's pretty clear here that this field is way less intensively drained than this group of fields, right? So here you have a couple of field ditches and there's a channelized stream here. Here you have ditch, 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 ditch. You know, it's all very, very heavily ditched, so we know that the drainage intensity is higher. When we look at things like tile drainage, we can actually calculate a drainage intensity because in a single field you're going to have multiple tiles. And in general, with the newer tile systems, they're going to be evenly spaced and they're going to be placed at about the same depth. So based on the spacing and the depth, we can calculate a drainage intensity. But what happens when you get into these ditch fields and the boundaries of your field are the actual drainage ditches? You can't really come up with a spacing, so what we had to do for our work was zoom out from the field and take an average spacing of those ditches and an average depth of those ditches over an area to kind of get a picture of how intensively drained something was. And the question that I have related to this, so how do we accurately account for this? Do you think the subsurface phosphorus risk from a low intensity grouping of fields is going to be the same as a high intensity grouping of fields? And I think no. But the way the PSI in Delaware accounts for subsurface risk, as soon as you get a ditch in your field, you are artificially drained and it pushes you to a high risk category. So we're not even accounting for that maybe you have a wet spot in your field, not, very poorly drained soils that need very intensive drainage. And so this is one of the issues that we look at, and how do we account for this when we move forward. So all these things, I hope I've explained how difficult this question is. What are we doing to improve our understanding? And this is where those two tools come in, the geophysical tool of electrical resistivity imaging and the hydrology tool of stable water isotopes. I'm going to spend my time talking about the first, and then I'm going to invite Lauren up here to talk about the second. This is my grad student, Katie Clark. This is partly her work from her master's degree. Some of you may recognize Katie. She now works for the Nature Conservancy up in Pennsylvania. What we did here is we applied this geophysical tool called electrical resistivity imaging. Who's heard of electrical conductivity or a Varus machine? I see a couple hands. So it's the same type of thing, except on a much more fine scale. So what we're doing is we put in these electrodes into the field at a specific spacing. We set them up to a box that creates an electrical current, and we inject the current into 2 electrodes at a time, and then we measure what's going on with the potential at the other electrodes. And based on this, we can make really cool pictures of the subsurface. And we can space these different ways so that we can look deeper, we can have better resolution or poorer resolution. So you can go deeper with poor resolution or shallower with really good resolution. And we have to kind of make all these decisions when we go out in the field. Ultimately, we can process this data, and here's in 2D. The folks we work with are really good at this, and they can process in 3D and give us models that we can turn around and look. Unfortunately, I only get to show you in 2D today. So we're doing all of our research at the University of Maryland Eastern Shore in Princess Anne. And the reason we're doing this is those are those field sites that the USDA was using and heavily has a lot of information from the early 2000s. So we wanted to start there because we kind of have an idea of what's going on in those fields. So you can see we're down here in Princess Anne. This used to be a field where we worked. They put a whole bunch of solar panels here. But we're here next to these poultry farms, and we're going to work in this field, but we're only going to work in small parts of this field. So here's Katie again, and here's our box that produces all the electrical current. And you can kind of see, we put some blue bars here to indicate the spacing of our electrodes. They're just metal rods. We have 192 of them that we bang into the soil about 2 inches deep. And then we hit them with electrical current before and during a storm event. We also have put a bunch of wells in the ground here so that we can kind of monitor what's going on with the soil temperature, or the water temperature and the water movement, the height of the water table, et cetera. So I can kind of show you that. Upstream of the ditch, we put in this little shallow trench. And something I should mention about this site is that about 10 inches below the soil surface or so, there is a really thick clay horizon that sits over this really great conductive sand. So we think water can perch on top of this clay layer. We think it needs to get into the sand and move out, but ultimately we want to see how is it moving. And what we did is we applied a salt in this little trench. So we dug this trench, filled it with the pea gravel, and then we pour our salt solution over it. We use potassium bromide because bromide is not really found in nature all that high occurring, so we can measure it in our water samples. And also it doesn't really stick to soils because it's negatively charged, so it will move with the water. So yes, it doesn't necessarily behave the same way as phosphorus, which we know is held by iron and aluminum in our soil. But what it does do is show us where the water's going. And clearly you have to know where the water's going to know where the nutrients are going. So we set this up and we collect data over time during different storm events. So we run the sample before, or we run the setup before it rains, and then we run it periodically about every 45 minutes through an entire rainstorm. And we collect these data. And our folks at Rutgers Newark can take these images and make these cool, pretty maps where they subtract what it looked like before the storm and only look at changes in conductivity. So anywhere that's red is highly conductive. Anywhere that's blue is even more conductive than what we had in the background. And we can make these pretty images, and we can look at how our tracer and the water are moving in the field. So this is another way we can look at it. I can show you over time. We can take slices. We can look at 3D. So that is where our trench was. We were looking cross-section here. Over time, after it started raining, you can see it gets really hot here with red as the salt starts dissolving in that rainwater and moving deeper into the soil profile. So like I said, we also have these wells. One is in the topsoil, one is in that sand soil. We're measuring water temperature, water height, knowing where the water table is during the year. We also pull samples from this and measure concentrations of salt so we can see if we have our bromide tracer moving. Potassium, look for bromide, and we also look for phosphorus in this water. So I'm going to show you 2 storm conditions. Our first storm happened in September, October 2015. where it was pretty dry out in the field where we started. And what you can see here is blue is the precipitation, red is our discharge in the ditch, and then these dotted lines are when we took these images. So these are differences from background condition, and anywhere you see color means that it's more conductive. So what we were seeing here is once it started to rain, we see it start to get a little bit more conductive in the surface. We're going down to about 4 meters here. This blue blob is indicating that our subsoil below that argillic horizon or that clay horizon is starting to get wet. And you can see here's our salt tracer getting really hot. And then we start to see this mass where it's slightly conductive. And it gets even bigger as we move. But when we look at this, we can measure and we can corroborate with our well data that the center of this mass, where there's some tracer, is moving into the field. So it was so dry that our water wasn't discharging to the ditch. It was actually going from the ditch into the field. So we were completely disconnected at this time of the year. And we had to wait for things to wet up before that water again discharged through the drainage system. So remember, dry conditions, disconnect, water moves into the field. Second storm event, we had a higher discharge. We've got some rain events here. Here's our same images. Here's our trench with all of the potassium bromide. And very quickly, this salt tracer found its way through some preferential flow pathways in that clay layer and got into the sand. And then it starts, as we move through time, moving toward the ditch. But what's interesting about this is that as it's moving toward the ditch, we can measure the time that it's taking for the center of mass to move. We are 4 feet from the ditch here. And we calculated that it would take about a year for that water to get to the ditch from that location. So that's very interesting to us because it's suggesting that we know phosphorus is going to interact with that soil and water as we move. So there's probably some sorption capacity that's going to remove some of that phosphorus as the water is moving. And that water is moving pretty slow, actually a lot slower than we thought. And so maybe we don't have to worry so much about what's 45, 50 feet into the field and away from the ditch. This is legacy phosphorus, not any manure that was put on. This is all based on soil phosphorus that was already there. So our conceptual model here is dry conditions, water moves into the field. Wet conditions, water moves out of the field and into the ditch, but pretty slowly. Okay, so now I'm going to invite Lauren to come up here and talk a little bit about what we're seeing with our stable isotope work.
Okay, hi everyone. I'm Amy's graduate student, and second part of this project is using stable water isotopes. And it's not as exciting as the electrical resistivity imaging, but it's still pretty cool. So first off, what is an isotope? So normally elements have the same number of protons as neutrons. With isotopes, that element now has the same number of protons but more neutrons, making it heavier. So we're just focusing on hydrogen and oxygen since they are the elements that make up a water molecule. So if you're looking at hydrogen, there's just one proton, but its isotope, deuterium, has one proton and one neutron. With oxygen, we have 8 protons, 8 neutrons, but its isotope, oxygen 18, has 8 protons and 10 neutrons. And if you have a cup of water, water molecules are naturally mixed with both hydrogen, oxygen, and its isotope. So this is naturally occurring. So if you have a puddle of water and it's a sunny day, water molecules that are lighter or have mostly just our hydrogen and our oxygen will be evaporated first, leaving behind our heavier water. So that's water molecules that contain mostly our deuterium or our hydrogen isotope and oxygen 18. If we're looking at clouds, the heavier water molecules or water molecules with our isotopes are going to fall out faster or first, leaving behind our lighter water molecules. So we can apply this principle of how water molecules are affected by environmental processes to figure out how phosphorus is moving through the subsurface. So as Amy said before, we can have water moving slowly through these small micropores carrying dissolved phosphorus. And water is sitting here a really long time in the soil because it's slow moving. So these water molecules are exposed to evaporation, and they're going to have a different weight or isotopic ratio than water that's moving really quickly through these large cracks in the soil carrying phosphorus. And what I mean by ratio, it's just comparing the number of hydrogen to deuterium and oxygen to oxygen 18. It's just counting in a water sample. And eventually, water will move from these subsurface pathways into a drainage ditch. And this is just a mixture of water from these two subsurface pathways, as well as some other sources such as precipitation and groundwater. So we can pick apart this mixture of water that's in our ditch to figure out, okay, what is the dominant subsurface pathway associated with phosphorus loss? So we're on the same field as before in Princess Anne, and I collected water samples for over a year from precipitation. We have our mobile water, which is just water moving through our preferential flow pathways. Our matrix flow water, also called bulk soil water from soil cores. Groundwater from piezometers, and then I'm also collecting water in the ditch, which is a mixture of all these sources during base flow, which just means non-storm event, and storm flow. And you can actually see we had a really big storm event in May that overflowed the ditch, and it's actually going on the road over here. These boxes are just our automatic samplers. Don't worry about those. Okay, so, oh, you took out that one slide. Okay, so using... It is? Wait. Oh, no, never mind. Okay, so collecting water samples all from a year, we can classify them into, right here we have water in the ditch during base flow, ditch during storm flow, groundwater, water precipitation, water moving through our preferential flow pathways, and our matrix flow water. And right off the bat, we can see that our matrix flow water, or that's moving really slowly through those micropores, is significantly different from our water in the ditch during a storm event. And we're really interested in that because that's when most of our dissolved phosphorus is moving through the subsurface. Looking at our preferential flow pathways, this is similar to the water in the ditch during a storm event. So just looking at these box plots with no phosphorus data attached to it, we can see that they're most likely preferential flow pathways or quick movement through the cracks is that dominant pathway feeding the drainage ditch. Soon we'll have dissolved phosphorus data to go along with each box plot, but as of right now, we don't. We can look at individual storm events through hydrograph separation. So this is one storm event in October 2017. So during a storm event, we can plot the discharge or the amount of water moving through a ditch at a certain amount of time, which is this solid gray line you can kind of see right there. Okay, and we can use a tracer, such as the isotopes that I mentioned before. So in this example, I'm using oxygen 18 as our tracer. So we're taking a water sample, analyzing it for that oxygen to oxygen 18 ratio, and we can determine the percent of event water, or this is water mostly moving through preferential flow pathways. And we're finding that after analyzing multiple storms and different tracers, we can also use deuterium and bromide and chloride, we're finding that at peak flow, so our peak discharge, event water, or water moving through preferential flow pathways, contributes 64 to 91% of the water in the drainage ditch. So that is that's showing it's the dominant pathway. Pairing this with the dissolved phosphorus data, which is this dotted line right here, This peaks, the concentration of dissolved phosphorus peaks at the same time as event water. So most likely we're having precipitation moving through these cracks within the soil, carrying that dissolved phosphorus that's stratified in that topsoil, and it's just moving through the soil. The same water that moves through these cracks might not be what ends up in the drainage ditch, as we're seeing with electrical resistivity imaging. It's taking a long time to move, but it's still starting that process of moving that phosphorus into the drainage ditch. So this was just a really quick overview of some of the results that we're finding. We just finished this study, so now we're looking at all the data.
Okay, so you might be saying right now, wait, Amy, you just told me that phosphorus and water move really slowly to the ditch, and Lauren just told us that it moves really fast to the ditch. So this is where we need to start connecting what we're seeing with these two types of studies, because we are seeing somewhat contradictory results. So how do we connect what we see at this subfield scale with what we are seeing at the ditch scale? Reminding you that it's kind of difficult when we look at the water isotopes in the ditch, because not all of the water that's discharged to the ditch came from that field. But just like any good scientist, we did our whole process backward. We went out and we picked a spot in the field to put this ERI grid, and then we already had this weir and everything from the previous studies for collecting our water samples. And so we just kind of guessed where we should put these things. We didn't really have any information about the entire field condition before that. So that's where some of these other types of tools, which we kind of call higher level geophysics, come in. This is a pulled electrode array. It's very similar to what I showed you with those electrodes in the ERI. It does exactly the same thing, except in this particular case, they've attached it to the mule or the gator, and they've driven it around the field to look at the field conditions at that time. We also can use electromagnetic induction sensors to map the subsurface. And then we have a Varus, so we ran the Varus on the field to also look at how the electrical conductivity of the soil was differing from what we were seeing with these other tools. And as we move forward with this work, we're hoping that we can start to generate maps of specific fields and the subsurface condition that would be useful to you as consultants and also to farmers when they start to make some decisions about conservation and management for their crops. So these field scale tools actually start to give us a more complete picture of what we're seeing. And so you remember I said that this site has a fairly thick clay horizon below the surface soil. What our map with our portable ERI says is that where it's red, and I apologize, they always put red, and I know a lot of people are red-green colorblind, and they just always put red in this scale. This is all red if you can't see it. And what this is saying is it's about, I'm really bad, it's about 4 feet thick, the argillic horizon. That clay horizon below the soil surface is about 4 feet thick in this area of the field. And it's less than a foot thick or even non-existent in this area of the field where you have a lot of blue. What's interesting is we ran that soil Varus over the same thing. We see a very similar pattern in the top foots. The electrical conductivity is much lower than it is on this side of the field than it is on this side of the field. So we are hoping we've convinced USDA that we've got some pretty cool tools. No one's going to be able to go out and put all those electrode arrays in a field and really understand what's going on, but these tools where we can drag them behind a gator and get a map, they could be really helpful. So why are we seeing these contradicting results? And we think it has a lot to do with this idea of critical source area in your field. If you went to Doug's talk, he talked about critical source areas with the P-index, same kind of concept. 80% of our site had the dominant soil condition. That's a typical quindaqua soil with that thick clay layer under the soil surface. But the other 20% was the minor soil condition where there was almost no clay layer to prevent the flow of water. And we think a lot of the discharge is actually happening in this region. And that's why we're seeing different results with the different soils. So as we move forward, we're wanting to take this concept out to additional fields. We actually have some soil core data from about 16 fields on Delmarva, and we're asking the USDA to fund us to go out and make these types of maps, because we're hoping that as consultants, you could take a map like this and say, you know, if we're talking about subsurface phosphorus loss, it's really this part of the field that we're the most concerned with. And so we need to manage it differently, or we need to maybe have a setback here, or figure out how to do something differently so that we prevent that phosphorus from moving, especially where we have legacy phosphorus in the first place. And right now, the way that our P-index works is it is based on the dominant soil condition. And so in this particular field, we would have based all of our information on this Quindaqua soil. It is all mapped as a Quindaqua soil because the level that you get from the soil survey is not fine enough. There is always some kind of minor component to the mapping units that you're seeing on those maps. So like I said, we are trying to identify some of these geophysics tools that can provide useful information for farmers. And what's nice about these is that it's unlikely to change in any amount of time that we'd be working the field. So once we map it once, you can use that map for pretty much as long as the grower is cropping that field. The other thing we're trying to do is the folks who are smarter than me, the hydrologists and the geophysicists, they've said, we think we found a way to use some of these tools to help us distinguish where we have matrix flow versus macropore flow, so that preferential flow versus that slower matrix flow. And we're hoping that with this information, we can tie it back to some of the common soil properties that we get in the soil survey and say, you know, these soil series are most susceptible to preferential flow conditions, which we can then use to help us make decisions as we move forward. So with that, just kind of summarizing what we found, we hope that we've shed a little bit of light on what's going on the subsurface. I think we've ended up with a lot more questions than we started with, but knowing that we're moving toward figuring this out, and hopefully by the time I retire, I'll have the final answer for you. But with that, I would be happy to take any questions. Okay, so the question was, with the information we're finding and knowing about legacy P, what kind of BMPs can we possibly use to mitigate this? Is that right? That is unfortunately not many. Legacy P is a really big issue. Cropping takes a long time. Nicole's got a really great paper in Agronomy Journal that shows that from different soil test levels, it could take 20, 30 years to crop that phosphorus down. Right now, we've been looking at different soil amendments to try to get more phosphorus into the crops. Sometimes looks promising, sometimes doesn't work so well. A lot of soil amendments actually convert phosphorus to a less available form, but that doesn't help us get rid of the phosphorus. It just changes the solubility and possibly in the short term. Right now, the ditch filters or treating that water seems like the only thing that's been really working, but there's a lot of issues, especially if we start thinking about these filters on a small scale. It's just like a water control structure. We put in water control structures. People are supposed to back the water up in the field during the wet season, and you see boards laying on the side of the field all the time. So it becomes a management issue. You got too many decisions to make, and you just don't have time to manage that filter. And so I don't have an easy answer. The best I can do is say that if we can identify these critical source areas, there may be something we can do to remediate those areas on a smaller scale rather than having to outfit every single high P soil with some kind of BMP to solve that problem. Okay, so the question was, how much difference in time is it between the preferential flow and the macropore and the matrix flow in our site? So if we look at that ERI site, the preferential flow through that argillic horizon, which remember can be up to 4 feet deep, is really quick. It's like a couple of days. And then it sits in those matrix pathways for a lot longer. We haven't been able to quantify that with the isotopes and on that part of the field where we don't have the clay, but we were just assuming that nothing moved through that clay horizon, but we found that there are cracks in it, and it is really quite a lot faster than we thought.